Hello everyone, welcome to my channel Anupma Biology Classes. In this biotechnology series, today our topic is tools of recombinant DNA technology. This video is taken from CBSE class 12th, part 2nd of chapter 11. Here we will know about the biological tools of the recombinant DNA synthesis. In previous video, we know about the recombinant DNA technology. Now, here you can see the lecture overview that what we will see in this video. So in this video, we will see tools of recombinant DNA technology in which enzymes, restriction endonucleases, separation and isolation of DNA fragments, cloning vectors, features required to facilitate cloning into a vector, passenger DNA and competent host. Now let's begin the topic. So at first, the question is how many tools are used in the recombinant DNA technology? So three types of biological tools are used in the synthesis of recombinant DNA, and these includes first one is enzymes, second is vacuole or vector DNA, and the third is passenger DNA. So these three tools are used in the formation of recombinant DNA. Now, the first tool, that is enzymes. So what are enzymes? So many kinds of specific enzymes are employed in genetic engineering. These include lysing enzymes, cleaving enzymes, synthesizing enzymes, joining enzymes, and the last is alkaline enzymes. So here we will discuss each and every part of these enzymes in detail in the next slide. So at first, the first enzyme is lysing enzyme. So what is lysing enzymes? These are used to open up the cells to get DNA for the genetic experiments. Lysozyme is commonly used to dissolve the bacterial cell wall. So, cell lysis is used in laboratories to break open cells and purify or further study their contents. Lysis in the laboratory may be affected by enzymes or detergents or other chaotropic agents. Lysis is also used for the protein purifications, DNA extraction and RNA extraction. Now, the second enzyme is cleaving enzymes. So, the role of cleaving enzyme is to use to break DNA molecule. So, the main cleaving enzyme is here, that is nucleases. Nucleases is of two types. First one is exonucleases, which cleave DNA in the terminal end. And the second is endonucleases, which cleave DNA in the middle region. Now further, this endonuclease is divided into two parts. First one, that is non-specific and may cleave DNA at random sites. And the second is highly specific and operate only at specific sites, and these are known as restriction endonucleases. So we will know about that what is exonuclease, what is endonuclease and what is restriction endonuclease in the next slide. Now you can see here the action of DNA cleaving enzymes. So in this picture you can see the role of exonucleases, endonucleases and restriction endonucleases. So at first, what is exonucleases? Exonucleases cut off nucleotides from fifth or third terminal end of the DNA molecule, which you can see in the picture. That means cutting or cleaving of a DNA molecule is at terminal position. Now second is endonucleases. Endonucleases cleave DNA duplex at any point except the terminal ends. And the third one, that is the restriction endonuclease. So what are restriction endonucleases? Restriction endonucleases cleave DNA duplex at a specific point in such a way that the single stranded free ends project from each fragment of the DNA duplex. These single-stranded free ends are called sticky ends because they can join similar complementary ends of the DNA fragments from some other sources. So this is the action of DNA cleaving enzyme, that is exonuclease at terminal, endonucleases at any point except terminal, and restriction endonucleases that cleaving DNA duplex at a specific point. Now, the third enzyme is synthesizing enzymes. So, these synthesizing enzymes play a role in the synthesis of DNA strands on suitable templates. And there are further two types, in which the first one is reverse transcriptase. Here in this picture you can see the role of reverse transcriptase, which help in the synthesis of complementary DNA strands on RNA template. And the second is DNA polymerases, which aid in the synthesis of complementary DNA strands on the DNA templates. So, DNA polymerase used as proofreading. So, the role of DNA polymerase is excises mismatches and continues synthesizing DNA fifth to third end. In case of prokaryote, DNA polymerase first and third are exonucleases. In case of eukaryotes, polymerase delta and polymerase eta are exonucleases. So, these are DNA polymerase and reverse transcriptase, which are synthesizing enzymes. Now, after this, the fourth enzyme is joining enzymes. So, what is joining enzymes? Joining enzymes help in sealing gaps in the DNA fragments which are otherwise joined by complementary base pairing. And the example in case of joining enzymes is TU ligases, which act as a molecular glue, and they join DNA fragments by forming phosphodiester bonds. Now, after this, the last fifth enzyme is alkaline phosphatases. So, what is the role of alkaline phosphatases in recombinant DNA technology? These cut off phosphate group from the fifth end of the linearized circular DNA to check its recircularization. So, this is the role of alkaline phosphatases and all enzymes. Now, the main enzyme, which is restriction endonuclease, which is also known as molecular scissor. So, what is restriction endonucleases? Restriction enzymes are isolated for the first time by Stewart Lean and Werner Arber in the year 1963. They isolated two enzymes which restricted the growth of bacteriophage in Bacterium E. coli. Out of these two enzymes, one was responsible for adding methyl group to DNA and the other enzyme was responsible for cutting or splicing the DNA molecule. The second enzyme was named as restriction endonucleases. The first restriction endonucleases, whose working depend on a particular nucleotide sequences, was isolated by H. O. Smith, K. W. Wilcox and T. J. Kelly in 1968. These enzymes was isolated from the bacterium Haemophilus influenzae and named as Hind2. It was later observed that Hind2 always cut DNA molecules at a specific point by identifying a specific sequence of 6 base pairs. These specific sequences of base pairs is known as the recognition sequence for the Hind2. Besides Hind2, more than 900 restriction enzymes are known today that have been isolated from over 230 strains of bacteria. Each of these enzymes recognize different recognition sequences. Now, here you can see that in case of restriction endonucleases, there are types of restriction endonucleases. So, in types, you can see here there are 3 types of restriction endonucleases, which are type 1, type 2 and type 3. RE indicates restriction endonucleases. So, according to property, that is in case of abundance, type 1 is less common than type 2 is most common and type 3 is rare. According to recognition site, type first cut both strands at a non-specific location, more than 1000 base pairs away from the recognition site single. While in case of type second, cut both strands at a specific, usually palindromic recognition site. And the type third is cleavage of one strand only 24 to 26 base pair downstream of the third recognition site. Now, according to property, that is restriction and modifications, type first is single multifunctional enzyme, type second is separate nucleases and methylase, and type third is separate enzymes sharing a common subunit. Now, according to cofactors, in type first, the cofactors are ATP, Mg second ions, SAM. According to second, that's Mg2 plus. According to type third, Mg2 plus, that is SAM, that is SAM. Properties, that is nuclease subunit structures. Type first is heterotrimer, type second is homodimer, and type third is heterodimer. According to DNA cleavage requirements, type first is two recognition sites in any orientations, type second is single recognition site, and type third is two recognition sites in a head-to-head orientation. Now, DNA cleavage. Then DNA translocation. In case of DNA translocation, in type first, yes, type second, no, and in type third, no. According to site of methylation, type first is at any recognition site, type second is at recognition site, and type third is at recognition site. According to enzymatic turnover, type first has no turnover, type second has turnover, and type third has enzymatic turnover occur. So, this is the comparative properties of the restriction enzymes. Now, here you can see that in case of type first, this is bifunctional enzyme, 3 subunits present, and recognition site bipartite and asymmetric. Restriction and methylation is type first is naturally exclusive, and cleavage site is non-specific, that is more than 1000 base pair from recognition site. In case of type second, separate endonuclease and methylases, recognition site is 4 to 6 base pair sequences, often palindromic. According to restriction and methylation, separate reactions occur, and the cleavage site is same as or close to recognition site. In case of type third, structure is bifunctional enzymes where the two subunits present. Recognition site is 5 to 7 base pair asymmetric sequences. Restriction and methylation is simultaneous, and the cleavage site is 24 to 26 base pair downstream of the recognition site. So these are the comparative view of all the types of restriction endonucleases. And here you can see the examples of the restriction enzymes, that is E. coli, that is organism sources Escherichia coli, TAC1, that is Thermus aquaticus, Hinther, that is Haemophilus influenzae, BAM-H1, that is Bacillus amyloliquefacens, and LU4, that is Erythrobacter luteus. And here you can see the recognized sequences, because you know that restriction endonucleases cut at specific sites. So these are the examples: microbial origin, enzymes, and the recognition site, and the cleavage products of the restriction endonucleases, which you can see in this picture. Now, after this, nomenclature of restriction endonucleases. So how can you give the name to any restriction endonuclease enzyme, like that if you talk about the Erythrobacter luteus microbial origin and the enzyme name is ALU first. So what is the way of nomenclature of this restriction endonuclease enzymes? So specific names of these enzymes are given by using the following conventions, in which the first one, the name of enzyme is derived from the name of the prokaryotic cell from which the enzyme is isolated. The first letter of the genus becomes the first letter of the name of enzyme. It is written in capital letter. The first two letters of the species makes second and third letter of the name of enzymes, and they are written in small letters. All these three letters are written in italics. The fourth letter of the name of enzyme is the first letter of a strain, it is written in capital. The Roman number written at the end of the name indicates the order in which the enzymes was isolated from the strain of the prokaryotic cell. So this is the way of nomenclature. Now here you can see the example. So like in case of enzyme E. coli, which is isolated from Escherichia coli RY13, and it is named as follow. That is, the capital letter E comes from Escherichia, that is genus. The letter co comes from coli, that is specific epithet or species name. The letter R comes from RY13, that is a strain, and the Roman number 1 indicates it was the first enzyme isolated from the bacterium E. coli RY13. So this is the way of nomenclature. Now you can see the second example, that is hint 3. So hint third is the Haemophilus, H genus name, IN that is specific epithet, D means strain, and the third means order of identification. So the name of here Haemophilus influenzae, here the name is hint third. Now what is the source of restriction enzymes? So bacteria are the source of restriction enzymes, and there are more than 900 known restrictions enzymes. So how are they named? The type of bacteria in which the enzyme is found, the order in which the restriction enzymes was identified and isolated, that examples are E. coli, are strain of E. coli bacteria, and the first, as it was the first E. coli restriction enzyme to be discovered. Now, what is the functioning of the restriction endonucleases? So, if we talk about the function of endonucleases, before this you can see here the applications of the restriction enzymes. Restriction enzymes are used in gene cloning and protein expression experiments. Restriction enzymes are used in biotechnology to cut DNA into smaller strands in order to study fragments, length differences among individuals, that is restriction fragment length polymorphism, that is RFLP. And each of these methods depend on the use of agarose gel electrophoresis for the separation of the DNA fragments. Now, use of restriction enzymes. Restriction enzymes are used in recombinant DNA technology, DNA sequencing, cDNA genome library construction, DNA mapping, and RFLP analysis. So, each restriction endonucleases inspects the DNA molecule in search of specific recognition sequences. Once it gets its specific recognition sequence, it binds to the site and cuts each of the two strands of the double helix at specific points by hydrolyzing the phosphodiester backbone. So here in this picture you can see that the recognition sequences, and the recognition sequence of each restriction nuclease consists of a definite number of specific base pairs in a DNA double helix. It is called palindromic nucleotide sequences. So what is palindromic nucleotide sequences, which we will see in the next slide. Before this, here you can see the steps in the formation of recombinant DNA by action of enzyme restriction endonuclease. So you can see here that is vector DNA and the foreign DNA, then the role of E. coli, then DNA fragments join at sticky ends, and the recombinant DNA formation occur. That means addition of vector DNA with the foreign DNA. So this is the whole action of the restriction enzymes. Now after this, here you can see the palindromic nucleotide sequences. So what is palindromic nucleotide sequences? The palindromes are the group of letters that form the same words when read either in forward or in backward direction. So here you can see that in fifth to third direction, that is T, T, A, G, C, A, C. So from the back direction, from fifth to third, T, T, A, G, C, A, C. So this shows the palindromic sequences. Palindromic nucleotide sequences in the DNA molecule is the sequence of base pairs that read same when orientation of reading is same. For example, here you can see the following sequences like AGCT and AGCT from both the from fifth to third direction in the both chain. So Palindromic sequences are of two types, the mirror-like palindrome or inverted repeat palindromes. So, the mirror-like palindromes in which the same forward and backwards are on a single strand of the DNA strand as in GTAATG, while in the inverted repeat palindromes is also a sequence that reads the same forward and backwards, but the forward and backward sequences are found in complementary DNA strands, that is GTATCAC being complementary to CATATG. So, inverted repeat palindromes are more common and have greater biological importance than mirror-like palindromes. The palindromes generally speak type second restriction enzymes. Recognition sites are the palindromes. A palindrome read from both sides yields the same sequence of characters. Examples are 1 to 1, IFF1, IB, ABA. However, for a DNA sequences, a palindrome refers to reading the sequences from both strands from fifth to third. For instance, the E. coli site is 5 GAATTC third, that is fifth to third end, and its complementary strand should also read fifth end GAATTC third end. So this is all about the palindromic sequence. Now, separation and isolation of DNA fragments. So how can we separate and isolate the DNA fragments? A molecule of DNA can be cut into fragments by the enzyme restriction endonucleases. These fragments of DNA can be separated by a technique of gel electrophoresis. Electrophoresis is a technique used for the separation of substances of different ionic properties. If such substances having different electric charges are taken in a solution, and if an electric current is passed through this solution, the charged particles also move either towards anode or cathode depending upon their charges, whereas positively charged molecules move towards cathode and negatively charged molecule moves towards anode through a medium or matrix. Since the DNA fragments are negatively charged molecules, they can be separated by allowing them to move towards the anode. The technique of electrophoresis was developed by A. Tiselius in 1973. Nowadays, the most commonly used matrix in gel electrophoresis is agarose, which is a polysaccharide extracted from seaweeds. DNA fragments move towards the anode according to their molecular size through the agarose gel. Thus, the smaller fragments move further away as compared to the larger fragments. The separated DNA fragments can be observed only after staining them with a solution of ethidium bromide. The bright orange color bands of the DNA can be seen only under UV light because pure DNA fragments cannot be seen under visible light. These bands of DNA fragments are cut out from the gel and extracted by using a convenient technique. This step is called elution. The eluted DNA fragments are then purified and then are used in constructing recombinant DNA by joining them with the cloning vectors. Now, the second biological tool is here, that is cloning vectors or vehicle DNA. The DNA used as a carrier for transferring a fragment of foreign DNA into a suitable host is called vehicle DNA. It is also called cloning vector or gene carrier. The desired gene is introduced into a vector wherein recombinant DNA, that is rDNA, is formed. The vector carrying rDNA divides, thereby forming several copies of rDNA. 5 types of DNA are used as vehicles, that is plasmid DNA, bacteriophage DNA, DNA of plant and animal viruses, transposons, and artificial DNA. So, the first two are more commonly used as vectors. So here, the two main types of cloning vectors, in which the first one is plasmid. Plasmids were discovered by William Hare and Joseph Lederberg in 1952 in the bacterial cells. They are small, double-stranded, closed circular, symbiotic DNA molecules that occur naturally in bacteria outside the bacterial chromosome. They are regarded as extrachromosomal DNA containing extrachromosomal genome. A bacterial cell may possess one or many copies of one or more plasmids. They are inherited from parent bacterial cell to the daughter cell and have capability to self-replication in the cytoplasm of the bacterial cell. The circular molecule of the DNA can be broken to yield a linear molecule which passes from one bacterial cell to another. There are many kinds of plasmid which differ from one another in size and in composition of genes. They vary in size from 2 kilobar to more than 400 kilobars. So this is the structure of the diagrammatic representation of E. coli, that is cloning vector here, that is PBR332 showing restriction sites, that is hint third, E. coli first, BAM H1, SAL1, PVU second, PST first, CLA first, origin of replication, that is V, and here the rope codes from the proteins involved in the replication of the plasmids. Now, the second is bacteriophage vectors. So, what is bacteriophage vectors? Bacteriophages are viruses that infect bacteria by introducing their DNA into the bacterial cells. The injected DNA molecules of the viruses integrated into the bacterial chromosome. This viral DNA multiplies with the bacterial chromosomes as prophages. The prophase may dissociate from the bacterial chromosome and assemble a number of phages which burst out of the cell and reinfect the neighboring cells, that is lytic pathway. This property of the bacteriophages to transfer their DNA into specific bacterial cells where they get integrated and multiply along with the bacterial chromosome gave scientists the idea that specially designed phage vectors would prove best tools for the gene cloning. Moreover, they are more advantageous than plasmid vectors because they can be used for off large DNA fragments and they can easily be detected at the time of cloning experiments. Now, after this, features required to facilitate the cloning into a vector. So what are the features which are required to cloning vector? So a cloning vector should have a relaxed origin of replication, that is ORI. This is the specific portion of the plasmid genome that serves as a start signal for the self-replication. This number can be artificially increased. Under natural condition, each plasmid replicates to produce 20 to 30 copies per cell. Now, a cloning vector requires the presence of a selectable marker to identify and eliminate non-transformants. It should selectively permit the growth of the transformants. Plasmids are characterized to possess some important genes like the antibiotic resistance gene, genes for production of toxic substances, genes for tumor formation, and gene for N2 fixation, etc. That means nitrogen fixation. These genes may act as selectable marker in transformations. A cloning vector requires a specific recognition sites where the enzyme restriction endonucleases make a cut so that a foreign DNA segment may be introduced to the vector. This property helps a vector to act as trusted cloning vehicle during gene transfer in genetic engineering. It is desirable that a vector must have very few, preferably single recognition site for the use by most common restriction enzyme. If it possesses more than one recognition sites, it will generate several fragments, which will complicate the gene cloning. However, presence of unique recognition sites for the several restriction enzymes within the limited part of the DNA provides flexibility in the choose of restriction enzymes used for a specific type of gene cloning. And the last is vectors for cloning genes in plants and animals. So here you can see a soil inhabiting plant pathogenic bacterium, that is Agrobacterium tumefaciens, infects broad-leaved crops including tomatoes, soybeans, sunflower and cotton, but not the cereals. It causes tumor called crown gall, which you can see in the picture, that disease. So, tumor formation is produced by its plasmid, which is therefore called TI plasmid, TI for tumor inducing. The TI plasmid integrates a segment of its DNA, termed tDNA, into the chromosomal DNA of its host plant cells. The tDNA causes tumors. As the gene transfer occurs without human effort, the bacterium is known as natural genetic engineer of the plants. Plant molecular biologists have started using TI plasmids as vector to transfer foreign genes of interest into the target plant cells. They use a version of the plasmid from which tumor-forming gene has been eliminated. The transformed bacteria do not cause disease. So, these are the features required to facilitate cloning into a vector, in which the first one is origin of replication, second is selectable marker, third is recognition sites, and the fourth is vector for cloning genes in plants and animals. Now, the third biological tool, which is passenger DNA. So, what is passenger DNA? It is the DNA which is transferred from one organism into another by combining it with the vacuole DNA. Three types of DNA are used as passengers. The first one is complementary or copy DNA, that is cDNA. Here in this picture you can see it is synthesized on the RNA template with the help of reverse transcriptase enzyme and necessary nucleotides. The DNA strands is isolated from the hybrid RNA DNA complex by using alkaline-phosphatase enzymes. A complementary DNA strand is then synthesized on the isolated single-stranded DNA template with the help of DNA polymerase. The cDNA duplex so formed can be joined to vacuole DNA for introduction into a host cell. So what is the way of formation of cDNA, which you can see in the pictures and all the steps. Now the second is synthetic DNA, that is sDNA. It is synthesized with the help of DNA polymerase on the DNA templates or from free deoxyribonucleotides without a template. So, now in case of synthetic DNA, there are two types. First one is artificial synthesis of DNA on template, which was introduced by the Kornberg and his co-workers in 1961. Synthesized DNA from the mixture of deoxyribonucleoside triphosphates, DNA polymerase enzymes, metal ions, and a segment of the viral DNA to act as a primer. So here you can see that the applications of synthetic DNA, and the second part, which is artificial synthesis of DNA without a template, which was given by Govind Khurana and his colleagues, produced the first synthetic DNA in 1965 by purely chemical means. They synthesized the gene coding for East alanine, that is tRNA, which needed only 77 base pairs. Unfortunately, these genes did not function. Kornberg, Khurana and his associates in 1979 synthesized the gene coding for tyrosine, that is tRNA of E. coli. Now, here you can see that the synthetic DNA application. So there are four basic applications: vaccine applications, diagnostic applications, drug development, and the basic science. So here you can see now the third part, that is the random DNA. It refers to the small fragments found by breaking a chromosome of an organism with the help of restriction endonucleases. So this is all about the passenger DNA. After this, what is competent host and what is the use of this in the transformation with the recombinant DNA? So, competent host cells, not every bacterial cell is able to take up plasmid DNA. Bacterial cells that can take up DNA from the environment are said to be competent. Can treat cells, that is electrical current and divalent cations, to increase the likelihood that DNA will be taken up. Two methods for the transforming, that is heat shock and electroporation. So, deoxyribonucleic acid, that is DNA, is a hydrophilic molecule and cannot pass directly through cell membrane. Therefore, certain treatments are made in host cells so that they become competent to take up DNA. Several such approaches have been made in the recent past to introduce the ligated DNA into the recipient cells. These includes chemical and physical treatment to facilitate the entry of desired DNA into the host cell. Some of these methods are here given below, in which the first one is chemical methods. DNA molecules can be forced to enter into the host genome only in those cells which do not possess cell wall. The naked plant protoplasts are mixed with the molecules of linearized plasmid DNA containing the foreign gene. The two are mixed in a transformation medium rich in magnesium ion and in place of calcium ion, following which 20% polyethylene glycol solution is added. After the treatment, the PEG, that is polyethylene glycol concentration, is reduced and the calcium ion concentration is enhanced. It promotes the frequency of transformation. The chemical methods involving transformation in presence of polyethylene glycol is convenient and simple, but there are however some disadvantages. So what are disadvantages? Many cells are so sensitive that the chemical method cannot be applied, whereas some cells die during the treatment. This method is not perfect because the treated cells do not contain any transfer DNA, and sometimes the fallen DNA is degraded into the cytoplasm before reaching the nucleus. Now, advantages of PEG. It enhances auto-abuse absorption. Pre-red cells treatment not necessary. Direct benefits are time saving and minimize the delay of urgent transfusion in via patient. Indirect benefits are the level and cost effectiveness. Disadvantages are the drawbacks of this technique relate to the availability of the suitable plant material and the inherent danger of the fibers which require careful handling. Many cereals produce embryonic callus that is hard and compact and are not easily transformed with this technique. Despite the some advantages, this method is recently used for the successful transformation of wheat, barley and maize without the need to cell suspension. Now, the second method of competent host is electroporation. This method involves suspension of plant protoplast in a suitable ionic solutions containing linearized recombinant plasmid DNA. This mixture is then exposed to low voltage long pulses or high voltage short pulses for the desired number of cycles. The electrical pulses are through the induced transient pores in the plasma lemma through which the DNA molecules are incorporated. Treated protoplasts are then cultured to obtain cell colonies and plants. This method is called electroporation. That means introduction of DNA into the plant cells by making minute pores in the plant cell membrane. Now after this, the next method is particle bombardment gun method. This method was first developed by Professor Stanford and co-workers at Cornell University, USA in 1987. The technique involves shooting foreign DNA into the plant cell at a very high velocity. This method is also known as ballistic technique. In this method, 1 to 2 micrometer gold or tungsten particles coated with the DNA are shooted into the plant cells using a helium pressure particle gun device. Important crop plants like wheat, rice and maize have now been transformed by this method. And the last is microinjection method. In this method, the DNA solution is injected directly inside the cell using capillary glass, micropipettes or microsyringe. This technique was originally used in the animal cells, but now it is applied in case of plant cells also. However, the method of microinjection required great technical skills because many cells die during the process. So, this is the end of video in which we know about the tools of biotechnology, that is enzymes, vector DNA and passenger DNA, and what is competent host. Now, the upcoming next video will be on the steps in recombinant DNA technology. That means how the formation of recombinant DNA technology occur. That means process of recombinant DNA technology. So if you understand this video, please like and share it and subscribe to my channel and my biology classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.